Hi, everyone. Thank you so much for coming, and good afternoon and good morning. My name is Meredith Price. I am the product manager for Imaris, and I just want to welcome you to our latest launch webinar. This is for Imaris 9.5, and the theme of this release is to tell your story even better with Imaris. And so really the entire team here at Imaris, that goes for the sales and support people that you interact with, as well as the development teams in Zurich and Belfast and the marketing teams as well. We're so excited for this release. We think that there's great features within this release that will mean a lot to you and in your daily work. And so we're excited to show it off to you, and we're excited to finally be able to get it into your hands after we've been able to play with it for a little while now. So within this release, we've made several improvements that should help your work stand out and be eye-catching, and to really allow your images to tell their story even better. And so we decided to make these improvements, these specific improvements, because we know that telling the story of your images and the story of your experiments is really like selling your own work. And so the success of your publications, your grants, your presentations is critically important to you, and your images are part of telling that story and are part of having a successful publication, grant, or presentation. And so the way that we are, PowerPoint, the way that we are thinking about this is that we want you to be able to easily generate results within Imaris. And so we've made improvements that will let you more easily generate a very specific type of application that we've seen a lot of our users perform, but also we want you to have a very nice image at the end that also helps you tell the story because it's easier to interpret. And so, for example, we have an image here. This is of a sea urchin embryo. We see some of the cells from it. We see that there is a green label here that is punctate, but also somewhat cytoplasmic, so we can make out the cells. We also have a nuclear label, and so you see that in blue. And you can imagine that for this data set, you might want to know where are those puncta in relation to the nucleus or in relation to the edge of the cell. And so performing that type of analysis is something that we wanted to go after as part of this release. And then we also wanted to go after presenting those results in a really nice way. And so hopefully you saw some images in the emails that we sent out inviting you to this webinar, and thank you again for attending. And so this is an example of an improvement we've made for surface rendering. This is a type of rendering that's included. We have a few different presets here. And what you're able to see now is that the segmentation for the cells, we've identified those. You're seeing them rendered as this transparent, glassy type of rendering. And we really like this rendering because it is so transparent that you're easily able to understand what's inside of your object, in this case cells, and to see the nuclei inside of it, as well as the puncta that have been identified. And so again, here, what we're seeing is that we're seeing the distance of these puncta from the edge of the cells. And so this is a very common measurement, measuring the distance of one type of object, so puncta, to another object, in this case, the edge of the cells, and understanding that relationship of how far away these two sets of populations are from one another. We see this over and over again. I just saw online on Twitter someone that posted a new publication where Imaris was used, and they did this exact type of analysis. And so what we've done within this release is to make it so much easier for you to do and so much faster, as well as to help you present it in a better way so that you can tell your story more easily and convey to your audience more about what your results mean rather than just what your results are. You can spend more time talking about your conclusions instead of just trying to point out what's in the image. So not only is this new rendering, which we're calling insightful rendering, there to be eye-catching, but again, it's there to help you really reveal your results to your audience in an easier way. And a lot of that is based around the improved transparency options that we're offering. And so you see that highlighted here in this image where we have a T cell interacting with a cancer cell, but also because of some additional information that we're able to include in the surface rendering, which I'll go over in just a second. So the first part of my talk will be about this insightful rendering. The second portion will be talking about these distance measurements that we're making, and we're calling it native because now everything is included inside of Imaris, and it's specifically included within our creation wizards, which is really this place where we find so much of the ease of use of Imaris is it comes from within these creation wizards because they do take you through the process of identifying and segmenting objects in your image very nicely. Also, because these distance measurements are found within the creation wizards, that also means that it's large data capable as well as batchable, and I'll go over that in more detail in that second section as well. And then finally, at the end of my presentation, I will talk about some other additions and other improvements that we've included in this release, and those, although smaller than these, are still incredibly important, and I know will put some smiles on a few customers' faces at least. Okay, so I'm going to turn back to insightful rendering because I want to go over what changes you'll see when you've downloaded the new version, or if you've not used Imaris before, how you'll interact with it and what it will look like so that you're not surprised. So really, we know that your story cannot depend completely on pretty pictures. And so this rendering is not just about having this rendering be very pretty, but also because it reveals more information. And so we're doing that through 10 different presets. And the first 4, which I've outlined in red here, and they're shown as spheres, that's the preview, are these spheres. These 4 are opaque, and I'm showing a couple of different versions, or a couple of different of the opaque presets down in the bottom left-hand corner. And so we see this first one shown here now. And so that is has some shininess to it. You do see some shadows. And then this option that I'm showing now is the third one. And so there's more shininess that you see. It gives you a different understanding and then a different aesthetic view of what this surface, what this T-cell is really looks like. We have 2 additional opaque options that I'm calling textured here because there is additional texture put onto the surface of the surface. This is not biological information. You can actually see it's in this blue cancer cell. But it can it's a nice addition to help you differentiate from one cell type to another, or one object type to another. And so that's something that you can also work with. And then finally, we do have 4 new transparent options. And this is where a lot of the revealing of what's inside and what's going on within your image can happen. And so these transparencies, these presets are made such that they work in many different places with many different images and applications, ton of time was put into making sure that these were foolproof for the vast majority of users so that they look great when anyone uses them on any type of fluorescent image that they have. And so here what you're able to see is this first transparent option, and you see that it's not completely transparent. There's more brightness around the edges, and we'll get back to that, but you can see through it, and you can see nice and clearly that this T cell is interacting with that cancer cell. And so you're starting to learn something about the biology because the rendering is able to show you that. And then I'm going to show you one more transparent type, and that's the one on the end. It's the glacier one, one of my favorites, because it does give you an even clearer look through, or a more transparent look through the surface. And so again, you'll have you have these options to play around with, and they do work in the vast majority of situations. So, but it's not just that these presets are have been well defined and that sort of thing. What they also do is that they've been made to have bright edges where you start to see the curve of a cell taking place. And so this example of right here of the T cell illustrates this, what we mean by bright edges. And so as the cell is no longer sort of flat on the top, but is starting to curve around down here towards the bottom, and as well along these filopodia that it's reaching out with, you see that the edges are very bright. You see that whiteness there. And what that is able to do, especially in a busy image when there's lots going on, is it's able to help you define what is in front of you, or what is closer to your eye, closer to the camera, and what's further away. And so it helps you to get that perspective within your image. And so you'll see that these bright edges are used in different ways throughout the different material presets, and they do add to the amount of information you can put in an image that helps convey what your results are to your audience. So the insightful rendering is available for surfaces. What you'll also see is that we have adjusted the color tab. So we're sitting in the surfaces right now, but we've also adjusted the color tab for the other objects, those being spots and filaments and cells, to match what it looks like here within surfaces. So now I want to show you how easy it is to play around with these, and how easy it is to switch things, and what it looks like within spots, the new color tab within spots as well. So I'm going to switch over to Imaris. I've got some images already loaded, and I'll show you what that looks like. So I have this image that we've been looking at. It's already loaded. You see the T cell over here on the left-hand side and the cancer cell over here on the right. When you finish going through a creation wizard within surfaces, what you will see is that when you then go to the color tab, you are presented with an interface that looks much like this. And so you'll have the 10 different presets, and it's very easy to just click through from one preset to another to find the one that either appeals to you aesthetically or helps you to tell your story better. And so in this case, it's having an opaque object doesn't tell the entire story here. All you can really see is that these two are touching each other, but you can't understand exactly how that's working. And so instead, what you might want to do is switch over to some of these that are transparent. And so now you can really see how the green cell is starting to maybe in the beginning stages of engulfing this cancer cell. And then you do have different options for those transparent renderings or different presets. One other thing that you may notice that's different on the color tab than before is that in the past the color tab was a hexagon, and now it is a line, or not, the color picker, sorry, was a hexagon, and now it's a line. And so you are able to either click straight to the color that you want, or you can drag the slider to find the color that you prefer. And so then you can pretty quickly and easily find the best fit for your image. You can also go in, I've hit this triangle and opened up this box down here. You can make it more saturated, you can add some gray to it, and so you can play around and adjust the color exactly to your liking by going into this view. Okay. So you have these options. Again, I want to point out that this cancer cell does have one of these textured options here, let's see. And so you do have both of the. You do have all of this available whenever you're rendering surfaces. And if you switch over and look at spots, so I've turned on these spots. They're foci within the T cell that were identified earlier. These the spots also have different material presets, and so this is the one that is most basic. You can also brighten it up a little bit with these two, and you also have a transparent option here. So just a little bit transparent so you can still see them, but you can make out where they are. Okay. So those are the changes that have gone into this insightful rendering. We are, as I've said before, so excited to let you all get your hands on this and start to work with this. We're excited to see future publications use this, and we think that these new transparency tools and the new renderings will really help you be able to show off your work without having to use maybe clipping planes as much or slicers as much, and so it'll be much more straightforward for you to export out snapshots and animations that illustrate the work you've done and the results that you have. So now I'm going to go back to the presentation, and then I will take you through the next story. So this next story is about our native distance measurements. And so we really wanted to make it so that we had a very easy method for our users, our new customers, to be able to understand not just the spatial organization of one group of cells or one type of organelle, but of the relationship of the organelles, maybe one type of organelle, the ER, relative to the mitochondria, or one population of cells relative to another population of cells, or to a reference object of some sort. And so we've seen that this is a type of work, an application that many of our users want to use and want to do, but it was cumbersome. And we found it cumbersome. We believe our users found it cumbersome as well. And so we wanted to allow this, these measurements and this assay to be as easy as possible for you. And so what we've done is to utilize the creation wizards. So, and I mentioned this before, and that's because we find the creation wizards to be such a nice way of going for attacking an image, essentially, and then generating your results. And so the vast majority of the time, when someone's using Imaris to do segmentation and object identification, they're using the creation wizards to do that in an automated way. And so it made sense to us to put those distance measurements within the creation wizards so that it's, again, easier to use, easier to train for all the core facility directors out there, to train new people starting in Imaris. And so that's what we've done. So I'm going to take you through that. And What it's going to give everyone, again, is an easy and automated way of calculating the distance between one population of objects and another. And it really speaks to the fact that we want to put the most common tasks within the creation wizards, because not only are they easy to use, but that's also how you will then go and do batch processing. And so by putting these distance measurements within the creation wizards, we're also allowing them to be included within batch processing. It also allows us to benefit off of the work we've done in the last 2 years, not only to improve batch, which was this past release in the spring, but also to benefit off of the work that we did to improve the analysis of large images with both surfaces and spots. And so you will see benefits there as well because of how we innovated and how we implemented this work within the creation wizards. Okay, so now I'm going to go through some of the different points that are important for understanding how this will these new measurements will be able to help you. So it's integrated into both spots and surfaces. When we're making a measurement with spots, we're going from the center or the centroid. When we're making measurements with surfaces, we're going from the edge. That's where the measurement starts and stops. And so again, you'll see that they're integrated in Creation Wizards. I'll show you that, and then when I go into Imaris, you see it at the very first step where there's a checkbox for turning on this shortest distance calculation. So within Imaris, we're calling it the shortest distance. That's the statistic as well, and that's where you'll find it. This came up in the previous webinar, so I changed my presentation for that. It also works within surfaces. You're not just going from the edge of a surface and out. You're also able to calculate things that are within a surface. And so I've already shown you an example of that, but you'll be able to do that as well. And again, it's because it's in the wizard, and we're calculating these measurements as we go, we can report the measurements within the filter tab and during the filter step of the creation wizard, so that you can use these as you're going along through the creation steps. As I mentioned before, they are included within batch, or this feature is included within batch, and it works with large images. And no additional software is required. So this is not no additional widget within Imaris is required. No additional MATLAB widget is required. It's all within Imaris, which means that it makes it much simpler for you to use, and especially new users of Imaris to begin doing this work. One note that I want to make sure I say to everyone is that we also realize that you can have many different components within one image, meaning many different types of spots and surfaces. And so because these are all calculations that need to be done, if you don't want to wait for the calculation to be done, you are able to turn these on and off during the creation wizard. You're able to turn them on and off afterwards as well. And we've made it so that only the first 4 components within your scene list, and I'll show you that in a little bit, have this automatically turned on. And so this should keep even more than most, the vast majority of people away from a situation where the calculations are taking a long time. Okay, and yes, this is the place within the Edit tab, which has the pencil icon, where you can turn the shortest distance calculations on and off if you find you don't need those. Okay, so now, I am going to get out of PowerPoint again and switch over to Imaris. And this time, I am going to take you through a data set where we have a nice neuron here that's been imaged. You see that in the red channel. And then you see many puncta. in the cyan channel. And so this, uh, so for this image, you can imagine that someone might want to know, well, I can create a surface to reconstruct the neuron, but what about the spots? And I want to know not where all the puncta are. I want to know just the number of puncta and where they are for the ones that are very close to this surface. And so what you can do is go through the steps. I'm going to turn this one off. Get a new spots object. Okay. So you can go through the steps of the creation wizard. I'm going to just switch channels to the cyan channel and put in the diameter I want to use to find all of these puncta. And it is making some calculations now, and now it's done. Awesome. So what it's showing me, first of all, let me zoom out on my histogram. It's showing me all the puncta within the entire image, and it's automatically thresholding to show me 52,000 out of 180,000 puncta that Imaris has detected. And but unfortunately, they're all throughout the image, and I just want to find the ones that are very close to the neuron. So to do that, what you can do within the creation wizard, and again, this is true if you are doing measuring from surfaces to surfaces, you can add in a filter at this filter step that's for shortest distance to surfaces. And so now, right now, it automatically is actually giving me just the spots that are 36 microns away, but instead I want the ones that are close. And so I'm going to move my slider over here to the left-hand side. By doing that, it actually turns off the lower threshold, and then I can right-click to turn on the upper threshold. And now you see that it's set at 5 microns. One thing I want to point out, I sort of inadvertently did it a second ago, is that you can zoom in to our histograms. So you could say, oh, there's a little bump there, and I could set my threshold right on the other side of that bump and see that that's at 0.576 microns. Instead, I'm going to move it out. I want it to be at 1, and then I'll just delete. Okay, so now instead of that 52,000 that were initially identified as good spots, now I'm down to 4,900. Okay, so I'm going to go ahead and finish the wizard. And so that's one application for using these distance measurements, is that it allows you to identify objects that are close and within a specific shortest distance range away from your reference object or from your set of reference objects. You can also go in now and color code those so that you can have an idea of where they really are in relation. So not just that they're within this band, but exactly where they are. So I'm going to statistically color code these, and I'm going to color code them again based on that shortest distance measurement. And so now within my image, I can very quickly get an idea of where these spots are. Are they inside the neuron? Are they outside? Are they farther away towards that 1 micron end? And so the ones that are purple and blue, they're inside the neuron. They have a negative. Let's see if I can move this out of the way. Yeah, you guys can see that through the WebEx. They have a negative value, and so that means they're inside of the surface, whereas the ones that have a positive value are going to be green and yellow and red, and they're outside the surface, and the red ones are about a micron away. And so again, you're able to have your image tell a story about what's happening within your experiment just from a simple snapshot, because not only do we have this nice rendering that gives you more information, but you also have the color coding and the distance measurements that help to describe what's going on within your image. And again, it's because we put these measurements within the Creation Wizards, they're incredibly easy to use, and I hope that many more people start to use them and are able to apply them within their research. Okay. So that's one example. Again, we did have the other example over here within the sea urchin embryo, where not only can we get the shortest distance from the edge of the cells, but I can switch it to where I'm using the reference to be surfaces 1, which is which are the nuclei. And so I can go into spots and change this. Oops, I need to turn that calculation back on. So I'm going to go into the Edit tab and turn that. So that's the pencil tab, and we can turn that calculation back on. When I go to spots, it did a really quick calculation. You probably didn't see it. And now that statistic is available within color coding. And so now I'm representing how far away each of those puncta that were identified as a spot, how far away they are from the edge of the edge of the closest nucleus. Okay. So again, this works for spots and surfaces. It's from the edge of the surfaces, it's from the centroid of the spots, and it's giving you the closest distance or the shortest distance from from an object in the other population. Okay. So I had mentioned that let me scroll through these. I had mentioned that it's also available with batch and with large images. So just for the sake of time, I'm not going to go over those two things in depth, but I do want you guys or everyone on the call to realize this and to try it out. You can also go to our website. So if you go to our homepage right now, it's imaris.com, you can click on the very top image, and it will take you to a web page where we have more examples on how to use this. We have short videos for those examples. We have different application types that you can look through, and then we have longer tutorials down at the bottom of the page to give you an idea of how you can apply this for your specific questions that you have. Okay. So we also have some other really nice features that are in here. And so the next one I want to talk about is volume rendering. Within Imaris, we do dynamic multi-resolution rendering of your raw data, so of your volume data. And we're able to do this because we're utilizing our IMS multi-resolution file format, and that gives us the ability to render the volume, or to render the portions of the image that we call resolution blocks or texture blocks, to render those portions that are closer to the camera or closer to you at a higher resolution than those that are farther away. So if you have a really big, let's say, whole embryo or whole organ, this makes a lot of sense because there's so much tissue closer to the camera when you're rotating and zooming in, and we want that part, the part that's easier to see, to be at a higher resolution than the parts that are farther away. And so we're making sure that we're optimally using the graphics card memory to show you the most important bits of the image. And so we're doing this dynamically, and I'm going to show you a movie now of how we're handling it now. What we noticed before is that there were some intensity transitions that weren't handled to our liking, and we wanted to improve those. And so now what you'll see is that in this movie that you're watching, the lower resolution is shown in green, and the higher resolution that's closer to us is shown in red. And so you're seeing that it's dynamically changing as this movie that Anna has made is rotating back and forth and zooming in and out. It's dynamically changing and adjusting so that however it's positioned, we're seeing the highest resolution when it's closest to us. And what our development team has done is to make sure that there's no sharp transitions from one resolution block to another in terms of intensity. And so you're seeing this blending of intensities across there. And so instead of just a sharp line between red and green, you're seeing bits of yellow in there. And so that really helps to make your final result of animations and snapshots very smooth and without any artifacts in there. And so we're very excited about this. We've already found it much easier for us to make very pretty movies like this for you guys to see. So it'll be nice to have that going out into the world and for all of you to use it. And then one other bit of improvement to the rendering of your raw data is when we're rendering data on 2D slices or within extended sections of our oblique or ortho slicers. And so what you'll see there is that the performance, so the speed of that rendering is much improved. And so if you had very large data and we're having some trouble with those ortho slicers and oblique slicers, you'll see that that's gone away now. So we're excited about that as well too. Okay. Again, so much of this release is really about enabling our users to have the best export, the best presentation of their images that we can allow them to have out of Imaris. And so our development team also spent time improving the animation exports. And so you'll see that those are improved. And then we also implemented something that we had done for snapshots or just the stills instead of the animations. And that's to make it so that you have a defined size that is then translated into what you see on the screen. So I have an entire screenshot here because I want you to see how there's gray bars on the left hand and on the right hand. So when you're exporting an animation or creating an animation, you know exactly what will be in that final result. And we find that when it's set up like this, you have much more control, and then you're much happier because you don't have to guess as to what will be in that final result. And so it really is what you see is what you get. And we also gave many different predetermined pixel sizes for the length and the width, or the height and the width of the screen for the animations, because over the course of time, our entire team, so I want to shout out to support here, found that using these specific sizes works better, not for the export necessarily, but really for the playing of the exported animations within the viewers. So that the video players want to have a specific size or a specific aspect ratio, and so we are giving those to you as defaults to make sure that the entire process for you, going from starting with an image that you load into Imaris all the way until you have to present it within PowerPoint, is as smooth as possible, and it gives you the highest quality result as possible as well. So we're excited to have everyone play with that too. Okay, then there's I am finishing soon, I promise. And so then there's a collection of other new things that are within this release too. We have, again, the color tabs have changed so that you have material presets and you have those spheres that give you a preview of what they'll look like. You'll see that in filaments, cells, and spots. And there we've also made some changes to how labels work. I'm not going over labels here. I should have made time in the slide for that. We've changed how they work a little bit on the color tab. It's going to be much more intuitive for current users as well as new users to jump into using labels. I would highly recommend that you play around with labels, and if you want to learn more, there are some tutorials online for that. You can get to those by going to our homepage and looking for the video tutorials. link, and that's at the top of the page. And you can also talk to your sales and support representatives to help you with those as well. We've included new demo images, and that's great for us because we're able to show off more of how Imaris works. We've included analyzed objects within there, so analyzed images with surfaces and spots. And I'm mentioning it here because I expect that there are some core facility managers and directors on the call, and I think these will help you when you have those first-time users that want to just play around a little bit, and you'll be able to give them some images that have already been analyzed so they can see what's what and get a better understanding of how Imaris works. We also have an ICS2 file reader. That had been something that the community had asked for. We're seeing that being used more often, and so that's now included. And again, you can you can convert those files over into Imaris files and then open them within Imaris. Python, the Python version support has also been improved. We are supporting both 2.7 as well as 3.7, importantly, along with this release, and so we're excited about that. And then in the vein of those within our community that are doing cell motility and tracking assays, we now have an extension for reporting mean squared displacement. So as a result of that extension, which you will need the XT module to have access to that, as well as for the Python support and interfacing with Python. But for the mean squared displacement extension, it will report both a plot and give you the raw data so that you can plot it elsewhere if you like. But you can get that quick plot out of the extension very easily. So again, there's a ton of different things within this release, but there is this theme of trying to help you tell your story better, because we know that you want to spend your time talking about how your results are affecting your hypothesis, and you want to discuss what those really mean, and you don't want to have to spend your time going through all the details of what you see in your image. So if we can help you tell that story better, we think we've done a good job and a good service for you. In addition to having those distance measurements done a lot faster for you and a lot more easily for you as well. So that is it for me in terms of presenting things. I do want to thank our customers and our users, because you do push us in the right direction for adding new features and making and innovating and providing new things like this awesome new rendering that we have. In terms of innovation, that's coming out of our development team. They are sitting in Zurich and Belfast, although they're probably home now because it's getting a little late over there. But again, we're taking the comments and the prodding and the pushing that we get from our customers and those that aren't our customers yet, and we're delivering that to our development team to have them think up the best way possible to implement and innovate to solve those problems for you. And they've done a great job, in particular, in this release, and we're really excited. I also have to thank Anna. She created a lot of the movies and images that you've seen on our website, as well as within the presentation, and she's done a wonderful job. You're going to hear her talking in the tutorial videos, and again, I think she does a great job of taking everyone through it. So if you have any questions, definitely check those out to get a head start so that you can dive right in once you've downloaded 9.5. The marketing team, thanks again for helping us push this out, and Sean, who's on the line right now. And then finally, the sales and support teams, who also push us. They're like our internal customers, and they have helped us improve the software since the beta releases, and I'm sure our customers love them as well. We have a really wonderful team, and I'm always very happy to work with each and every one of them. One last thing, we will be at SFN, and I think our booth is 1625. And if a Imaris team member wants to correct me, that would be fabulous. But come see us at SFN. You can find us. You can search for us in the app. We will be showing off 9.5, so come visit us. You can learn more there and chat with sales reps as well as some of the support team members that will be there. So I encourage you to do that. And that is definitely it for me. And there's both a chat box and a Q&A box, and you can start answering some questions over there. I'm going to stop sharing my screen, and I will answer any questions that I get. But it seems quiet. Okay. Okay, so one question was from Vitali. Hi, Vitali. You're wondering where the 0 is within the spectrum lookup table. And so you can set the minimum to be 0, and that would be the best thing that I would recommend in that case. Okay. And if you do have questions that you would like answers to and you've not already hooked up with your local sales representative or support team, feel free to email me. You've got my email address from where we sent out the webinar announcement. So go ahead and send those to me, and either I can answer you directly or get you to the right person to take care of you. I did not show vantage plots, but those are possible. These shortest distance measurements are used just like any other measurement or what we call statistic within Imaris, and so you can use them just like you would with others. Okay, so Karen is asking how is the shortest distance measurement being calculated with respect to surfaces? And so it is using the edge. It's not using a point within the surface. As you mentioned, it is using the edge. Okay. So then also these distances are available if you are measuring from surfaces to surfaces, surfaces to spots, spots to surfaces, or spots to spots. So there's 4 different combinations that you can use there. Okay, so yes. So Elena is asking, I hope I pronounced your name correctly, apologies if I didn't, is asking if you're able to export this rendering data to reconstruct it out to Imaris or outside of Imaris, and there are some steps for doing that. So I am going to, after this is over, I will send you some information about how to do that, and we will make sure to get that up on the web as well. So there is a way. Okay. So Ben is asking if the MATLAB distance transformation will still, that extension, if that'll still be available through the XT module. And it will. So there are other applications that this measurement isn't able to address. And I'm guessing Ben realizes this. And so we're not going to get rid of that extension because there's some extra things that you can do with that. It's definitely something that we're interested in for the future, not getting rid of it, but incorporating more of this type of work. And so you will still be able to create a distance channel and use it from there, but we found that just getting some of these measurements out was used so often, we wanted to make it easier for people. Okay, how am I doing? Let's see. Sorry, it's a very tiny box and a good number of questions. So I'm trying to grab all of them. Okay, I do think that's it. I hope everyone was able to get their sound taken care of in the beginning. If not, um, we will have a recording of either this version or the previous version that I did earlier this morning. We'll have that up on the website within a couple of days, perhaps probably even tomorrow, and so you'll be able to watch that. If you have any other questions, again, everyone should have my email address. I'm getting all of the auto replies. So feel free to just go ahead and shoot me an email. And again, if I don't have the answer, I'll find someone that does. And I want to thank everyone for coming and for great questions. That was a nice end to the webinar. Come see us at SFN, and yeah, let us know how everything works out for you. If you're interested in a demo, again, you can go to our website, click on the 9.5 image, and you'll be taken to a place where you can request a demo version of the software so you can try it out for yourself. But yeah, thanks so much, and we look forward to talking to you soon. Take care.